PNS nerve, poked in the eye, lose your sight, wait 3 or 4 years, allow time for the nerve to grow back, and you have your sight back. That's why we can't do this. Even though my eyes, and anybody else that's an organ donor here, may have put on their card their eyes to donate, as of right now, no one will ever get your eye in their socket to be able to see from it. Because we can't do this. Cut the optic nerve, take one eye out, put a donated eye back in, and have the optic nerve grow together. It's a CNS nerve. It doesn't have a Schwann cell, so there'll be no regeneration. So why do you donate your eyes? What part do they take? The cornea, the clear outer covering, which can be lifted off, put onto another eye, stitched in place, and the cornea is the thing that focuses your light rays coming in, the main focuser. So you could change a person's vision by improving their cornea. Question? They don't necessarily have to grow back the same pathways. They don't have to grow back to the complete form they would before. They can grow less or more. PNS nerve grows when it's damaged about 1 millimeter a week. So 1 millimeter is tiny. A little space in between my fingers here now. So if you sever a nerve up here, it may take years for you to get the feeling back in your fingers. I have a friend, and you'll see now that he's careless. I might have told you about it. He cut fingers off his hands. In fact, he cut 7 fingers off of one hand. 7. Cut those 4 on an angle, pushing them through a bandsaw. They were sewn back on. Then a year later, he cut 3 of the same 4 off again and sewn back on. So his nerve regeneration in his hand is a wreck right now, and it's been so 5 or 6 years. What little growth he got back, he cut off again and damaged them all over the place. And right now you could take a needle and press it straight through the end of his finger and he wouldn't feel it. He's got to be careful. He can't put his hand near hot things because he can burn his hand and not even feel it. He's still a carpenter, but he's a little bit more careful, I hope. So nerve regeneration takes place because of the Schwann cell. Take a look at the picture that I handed out for you. You're looking at the Schwann cell in this view, in cross-sectional view. We need some letters here on this. Let's call the top diagram A, the middle diagram B, and the bottom diagram C. The top is embryonic. A is an embryonic view of a Schwann cell. B would be a fetal view of a Schwann cell. In other words, later in embryonic development. And C would be a good example of a mature Schwann cell. This would be as a newborn. Find the axon in the picture. It's dead square in the center of picture A of the Schwann cell. The big rectangular shaped structure in there is the nucleus of the Schwann cell. Here's the nucleus right here, showing it to you in another view. It's like a little bump on the outside. This, by the way, is one of the things you got to identify in lab this week. You've got to take this model and look for the parts. So I'm giving some of them to you right now. One Schwann cell here is about this long from index finger to index finger, and is formed like this. Take a look at A, B, and C and watch what happens. It wraps and wraps and wraps, layer upon layer upon layer around the axon. The more wrapping we have, the more insulation we have. What is this building up on the outside of the axon? A coating. The thicker it is, the better it is, because that way 2 axons can't lie over top of one another and do what? If there was no Schwann cell on the outside. What would happen if you had 2 bare electrical wires and laid one on top of the other? It'd cause a what? A short circuit. You would have one signal from one wire jumping onto the signal of the other wire and causing a backfire. You wouldn't have any messages being sent right. Now, if you take those two wires and put plastic around them, dip them in plastic, you can take the two wires and put them right over top of one another, and one electrical signal stays in one and another electrical signal stays in the other. That's exactly what's happened here. The wrapping of the Schwann cell, forming layer upon layer upon layer, as you see in picture C, prevents information from going from one axon to another. I want to spend the time there on picture C. The fine wrappings of the cell membrane form what is called the myelin sheath. That is labeled for you down at the bottom. And what it amounts to is fatty insulation around the axon. And I like you to use the word insulation because that tells you exactly what it's doing for the axon itself. Notice that it is not tight against the axon. There's a little tiny space, and I've labeled it for you, called an extracellular space between the axon and the myelin sheath. That area is filled with liquid and ions. And that area has to exist in order for an impulse to go down the axon. Ions and liquid. The liquid is water. Now, look at one other label on picture C. Way to the outside of the Schwann cell, the last layer is a thin layer called the neurolemma. I think I've labeled it up around 2 o'clock on the picture. It is the neurolemma, the outermost covering of the Schwann cell that has the powers of regeneration built into it. That's the secret. That's the one that allows the CNS nerves not to regenerate and allows the PNS nerves to regenerate. Why? CNS nerves don't have a neurolemma. Their cells don't form that outer thin membrane that's important for regeneration. Ultra important, put a star next to it. When we talk about nerve regeneration, that's going to be the whole ball of wax, the whole saying, the whole important part, neurolemma. What cells have it? PNS. Where one Schwann cell ends, take a look up here, a new Schwann cell begins. My 2 index fingers show a little dip, a little indentation. That indentation is the beginning of one Schwann cell and the end of the next. So the entire axon is covered with Schwann cells. My hand is around one Schwann cell area. My hand's around the second Schwann cell area. Now there's some gray stuff on the outside. In lab you're going to find out that's just a little protective connective tissue layer. But the important insulation is the Schwann cell. Questions on it? This is called then a myelinated fiber. If you have a myelin sheath around an axon, it is referred to as a myelinated fiber. Now in lab this week, one of the pictures you're going to have handed out to you from the yellow histology book is going to be about myelinated and non-myelinated fibers. Every nerve fiber in the human body has got a myelin sheath around it. Every single one, unless you're diseased. Some diseases cause the myelin sheaths to disintegrate. That means all the signals get jumbled up. But in a normal, non-diseased human being, every fiber has a myelin sheath. How then could we have something called a non-myelinated fiber? Kind of a misnomer, I guess. Here's what you have in your picture there. with the axon in the middle. And here's the rest of the cell. And there's the nucleus. A myelinated fiber then has one axon for one Schwann cell. Here is what a non-myelinated fiber is. Without a whole bunch of wrappers, matter of fact, with just one wrap around each axon, nucleus is in the middle, and each one of these little yellow dots I'm putting in is a small axon. And so this Schwann cell here has gone and protected 1, 2, 3, 4, 5, 6, 7, 8, 9 different axons at the same time. Instead of it being wrapped around them, it just forms a little groove that it lets the fiber run through on. Is there a myelin sheath around it? A little one. Is it non-myelinated? Not technically. But by definition, we say that's a non-myelinated fiber, and that's a myelinated one. That's totally insulated. This is insulated a little bit. But my original statement holds true. Every single fiber in the human body has a myelin sheath around it, to some degree. Questions on Schwann cells? Example number 5. Who's my clock watcher today? How close are we? Anybody know the time? 4:15? Okay, it says here 11:47. What time you got back there on that clock, Brian? You got 11:48 now? Okay. We'll just wait for the bell. Example number 5. Get this name down, and if we get interrupted by the bell here, we're still pretty close. Called oligodendroglea cell. OLIGODENDROGLIA. Yep. OLIGODENDROGLIA. Oligodendroglea, second word, cell. And you need the goldenrod picture to take a look at this guy. And we'll look at it in 4 minutes, people. Break now. All righty, people. Everybody take this picture, please. Goldenrod picture. Okay, this is example number 5. It's called oligodendroglea cell. And if you'll find that label right dead square in the center of the page, I'll very quickly here make a long story into a short one with this, because you, I hope, understand already what the Schwann cell is. This is the central nervous system version of a Schwann cell. This is what takes the place of the Schwann cell in the brain and in the spinal cord. Instead of one cell wrapping around one axon in one little region like we find in the PNS, somebody take a look at that picture and tell me how the oligodendroglea cell is different. What's it do that's different than the Schwann cell? What's it do that's similar to the Schwann cell? The picture kind of tells it all. Anyone? Yeah, Sarah.
Well, it wraps around a lot of different ones.
How does it do that? What's it got to form?
The pseudopods?
Number 1, the oligodendroglea cell forms 20 to 40 pseudopods.